Bacteria colonize all types of environment on the surface of Earth. For example, they colonize plants and also human bodies in very large numbers. In order to colonize these diverse environments, each species has evolved different types of modules that are encoded genetically, allowing them to adapt and really compete with different species in this specific microenvironment. But we can actually harness these different modules and engineer them in synthetic bacteria in a way that will allow them to perform specific tasks in desired conditions. And for example, now we can envision using different tools engineered into a single bacterium that could be used to treat different diseases. For example, in a bacterium that combines sensing components of disease tissue, adhesion components to the disease tissue, and then treatment components that includes the secretion of toxins to kill the tumor cells or to the delivery of other effectors. With the help of the Gebert Rüf Stiftung, we are aiming at developing such therapy that would eradicate specifically cancer cells upon sensing the tissue using a sense of touch that we would engineer in the bacterium, and that would comprise adhesion molecules that would help the cells specifically attach to a disease tissue.
We engineered bacteria using a Velcro-like system. To do so, we displayed nanobodies at the surface of bacteria. They consist in small antibodies from camels and llama, and they are very modular. We can basically target any disease biomarker. And we improved the binding using time-lapse microscopy and microfluidics.
Altogether, we were able to design a bacterial system that has the ability to bind very efficiently and robustly to specific tissue. In fact, we believe that this system will be widely applicable to all kinds of bacterial-based therapies and even cell therapies to increase their specificity. In the future, we will seek to actually generate a real bacterial therapeutic that leverages this Velcro-like module by combining it with a sensing system that will allow the single bacterial cells to sense the presence of a tumor, and with toxin secretion that will help the cells eradicate this disease tissue.